Thank you. And thank you all for inviting. And yes, my name is Jürgen FRITZ. I work at university in Kassel and also at university in Bonn. And I worked in biodynamic agriculture about 7 years, and I had a lot of questions to understand the biodynamic background. That was the reason that I started at university, and since more than 20 years, I make experiments with the biodynamic preparations. So for me, it's not easy to switch directly in English when I talked the time German. So I need maybe a little bit time, but we start, no problem.
We have an agreement here that the common language is called broken English.
Broken English, that's good.
Yes, if anybody would come from South Africa or from the UK, they need to break their language. So please feel at home.
They're very good. Okay, then I can begin. Yes, I can open. Do you see the picture?
No, it did not come up at all.
Okay. You have given it free?
Yeah, I give you a share screen, but...
Okay, I have it. I think I gave it.
Yeah,
you have it now? Okay.
But we see your screen, it's not present mode yet.
Yes. I start here. Okay, and I will make in between, I will make a break so that we can talk a little bit and then I continue. And wait, I must. Okay, that's good. Okay, I want to talk about the influence of the biodynamic preparations on the plant growth promoting microorganism. So about bacteria and fungi in soil. And yeah, normally we say that is plant growth promoting microorganisms. I make a small introduction about this. And then I show our results from experiments. And then we make conclusions and continue. And you can ask in between if you have a question of you understand something, no problem. So we are a small group. Okay, we have big problems with human-made climate change. And this makes extremely events, growth stress, dryness, wetness, heat, cold, all this, of course. And one player of one thing we can do, and it's planned of its one important part in agriculture are the plant growth promoting microorganisms. The microorganisms in soil, they have a big influence of the health from the plant and how they grow, and also how they can act against stress. So it's expected for the following 20 years that this part with the plant growth promoting microorganisms gets more important than the normal agriculture, chemie and what they are spraying and the cost about this. So it's expected that this is growing very strong, this part. And one thing is very good because it's environmental friendly and resource saving. That's easy. But there's a big problem. The success of colonization is very difficult. And we will talk about this. So plant growth promoting microorganism, the inoculation from soil is not good if you have only individual strains. So it's much better. It is much better when you have consortia. So several harmonized strains. At university in Bonn, they made a work about several strains who work together, but it would be much better if we have more. So normally the origin, when they make an inoculation with plant growth promoting microorganisms, the origin is normally laboratory. And they say, oh, it's much better adapted if we take it from nature. But yes, 4 parts they do is they isolate from nature and then they continue. And yeah, how then the question is how to multiplicate, and mostly normally they do it only in laboratory and not in natural context. Now, when we look at this, and it's difficult to get it good, inoculate the plant growth promoting microorganisms in soil. So what can we do to make it better? So one part, the best one would, when we have consortia who have not only 5 strains, but maybe 300 or something like this, who are concerted.
Just 2 minutes, a small question maybe.
Yes, of course.
Jürgen?
Yes.
Are you talking about a normal habit of farmers to inoculate the land or the soil with bacterium?
Yes, that's what they are doing in normal farmers, what they are doing, and what they expect the future is from plant to help the plants to grow. And this is expected.
It's a new kind of product called biostimulants in general that you refer to, right?
Yes. That's biostimulant, and you use not big amounts, but you use greater amounts on soil we know that the microbiome in soil is very important for the growth from the plants. So, and there you try to bring it in a good mood, in a good situation to help the plants to grow. You understand this? And yes, biostimulants. But we change very quick to the biodynamic preparations, don't worry. So normally, it would be good to have concerted rhizosphere communities. It would be good to take it from nature, not from laboratory, the bacteria and fungi. And it would be good to take it from the natural context to increase them there. And now when we look, what we see here in the green parts, it arises the question, if we don't have exactly what the best would be to make an inoculation, the biodynamic preparations. Because we have a concerted rhizosphere community, it's very good adapted. We make it in the ground to produce it. We have a natural context, and we have, we fermented it in soil, so in natural context. And the big question from the lecture now is, of what I talk about is, are the biodynamic preparations a plant growth promoting inoculation? Do we put bacteria and fungi in soil as an inoculation so they can grow there and help the plants to grow? Okay. This is now the hypothesis for the next parts. When we look what is still now published about the explantation of the effects of the biodynamic preparations, there are a lot of literature about hormonal effects. So horn manure has auxin, it's one of the hormones producing bacteria. We know that signaling molecules could be interesting, and there is also published about this. And we know that networking for microbiome is better with biodynamic preparations than without. So we know from the background how the biodynamic preparations work. We know that the microbiome is important. But now we start with our hypothesis. The hypothesis is biodynamic preparations are an inoculation with plant growth promoting microorganisms. We will look about this. And And the first step is that we look what is in the biodynamic preparations. Do we have plant growth promoting microorganisms in there? And when we have a look now, we see different groups. So we have 3 hormones. They help to grow the plants to grow stronger. It's auxin, cytokinin, gibberellin. And the bacteria and fungi, they produce these hormones and help the plant to grow by doing this. And there are bacteria and fungi who produce stress, who help the plants for stress adaptation. So what can we take? If we take EPS, this is something colloidal in soil. It helps to make structure in soil, and it helps against growing stress if you have it heat or wet or something like this. Okay. And you have also bacteria who produces substances against salt, who help salt tolerance. Okay, you have different.
So in this table, you show that in the preparation themselves, there are bacteria that are making these materials. Yes.
Yes, the bacteria and fungi, they produce these substances who help the plant to grow better.
Yeah,
yes.
The preparations are diluted in water. So after you dilute it, you don't have the bacteria anymore, or what?
Of course, we expect that you have the bacteria and fungi, you have activate them. Normally you have only 1% active, and 50 or 80% you have dormant. They are not really active. They are close to active, but they are closely but not active. And we expect when we take warm water and we steer 1 hour, we expect that we activate from 1% to 50 or 60% that they get active and are no more dormant. So this is one part. So make questions in between. This helps. Otherwise, I don't get you in the talk. So
yep, I've got
very
one.
Good. Yes, very good.
In the text you see in the table we are looking right now,
Yes.
there is a considerable difference between 500 and 500p. Is this something which has to do with the region, or it has to do with the results of the analysis?
Yes, both are working, 500 and 500p. We have both with good figures in it. And we have good results in Cluny, that is the preparations from Masson. But also the other preparations are doing well. And we see that with on silica, the percent is much higher, but we expect there is not a lot in, the amount is not so high. But we see in all preparations, 500, 500p and on silica, we have high amounts of plant growth promoting microorganisms. This is the important first part. What is the question of? Otherwise, ask again. Maybe I don't know it.
Yes, I think you answer, but one more is coming. How you interpret the differences? I mean, in 501, for instance, there is double or triple times. Is it something which has to do with the initial substance, with the technique, or with the region, or both?
We expect not the region. We expect on silica has a higher percentage of plant growth promoting microorganisms, but we have not so high amounts of bacteria and fungi. Most is on silica, so we expect the amount is not very high, absolutely, but the percentage is very high. So maybe we get an important impulse on the plants with this. And we see it later that when we are spraying on silica, that 6 hours after spraying, the wine leaves get another structure in secondary ingredients. I show you later. But so that we get very quick an impulse for plants and for secondary ingredients. Yes?
Jürgen, I remember from India, from Savdaman, not Savdaman Fatal, I remember the research name later. They did a gene profiling of the microorganism in 500 and 501. And
Yes.
you could literally see that they are complementary. If you have one dial, you have this circular form of the profiling, and
Yes.
the one in 501 are different, completely different than the one of 500.
Yes, of course, they are really different.
That could lead to the inference, to the difference that you see here in the quantities, because it's in a sense another region of activity.
Yes. It's really different. Of course, we expect also this. So cow manure and silica is, of course, very, very different in bacteria, in fungi. Yes. Okay. So the first step is high amounts of plant growth promoting microorganisms in the preparations. And we have this. You can see it here. Okay, then the next step is to look, okay, when we put the preparations in field trials on the field, do you have then more plant growth promoting microorganisms in the soils where we put the preparations on? This is the second step for our hypothesis that the biodynamic preparations are plant growth promoting microorganisms and inoculation with this. Okay, we look here. We look in Frankenhausen. Frankenhausen is a university farm, and we have 16 pairs where we looked with and without preparations. And you see the green ones are the soils of the variants without preparations, and the red ones are with preparations. And you see overall the phytohormones, it's significant higher with biodynamic preparations. And all the preparations are significant higher with spraying the preparations. The soils also have significant higher amounts of plant growth promoting microorganisms. And you see the same with stress adaptation. The whole group, everywhere it's higher with biodynamic preparations. The plant growth promoting microorganisms are higher, and mostly they are significant. We see this in Frankenhausen, and we see this the same also in France. So we have checked here about 21 places all over France, and about 13 were wine growers, and the rest were agriculture, normal agriculture with different fruits on it. So it's very, very widespread soil and situations, and what we see is the same. So when we have all plant hormones, it's higher, significant higher with biodynamic preparations, plant growth microorganisms in soil. And you see it over all
exactly
groups.
did you check, Jürgen? Did you check the quantity of the bacteria, or what exactly did you check in the soil?
The quantity of the bacteria. The quantity of the bacteria, and yes, this is what we are checked. And a laboratory in Spain are checking this. So you really need specialists for this. So, and it's really special. But we all get it over 21 places in France. We also get the same result. And then we have an experiment in Darmstadt. And there we check, blue is 1 time on manure and on silica. Red is 3 times on manure and on silica. So you have an experiment about intensivation. Normally in Germany, it's only need that you put it one time each. And the question from the farmers is often, okay, I put it one time, but it's interesting to put it more, or it makes it no difference. And what we see is
one time a year or one time for the grow?
One time the year.
A year.
And if you have salad, normally one time per culture. But I don't know if everybody do this. But not one time a year, yes. And here, yes.
Can I have another question, please? Sorry to interrupt you.
It's nice if you ask.
After how long did you see the results? Was it like immediate, or a couple of months, a couple of years of implanting the preparations? Like how fast the results were obvious, let's say? Or when did you take the sample after the introducing for the first time the preparations?
I show you later that we made the chief from the laboratory. He says, oh, if you expect an inoculation, then you must make a time series and look after before you spray it, and then 2 weeks after, 4 weeks after, and you expect that the effect gets higher, still a point, and then it goes back down again. And we will look this later. Okay. Okay, but the intensivation in Darmstadt, we see the intensivation has an effect, and he is significant, high significant. So we have very good effects here also. And then we have another one, an experiment in Geisenheim. It's about wine grower. And here we see, first, we have in Darmstadt, we have 16 pairs. In Frankenhausen, 16 pairs. In France, 21 pairs. In Geisenheim, we have only 4 pairs, the basic for the statistic, and we have no significance here. But what we see is that it always goes down with preparations. So we get less plant growth promoting microorganisms in Geisenheim when we spray the preparations. And Geisenheim is the only place I know of experiments I know where the conventional variant has more nitrogen than two organics, organic and biodynamic. So you have there, how do you say, with legumineuses, you have high growth, it's about 1 meter. They let it grow every second row. In between the rows, every second year, it grows 1 meter high, and they work it in the ground. So they have a lot of nitrogen and a lot of substances for the plant. The level I think is too high for the growth of the plants. So if you look at the preparations as regulating the plant growth, and we expect that, or we know that the plant in their roots, or clothe their roots, they make more the bacteria and fungi that they need. And I expect in Geisenheim they have enough nitrogen to grow, so they don't push the bacteria and fungi who make more growth. And therefore I think this is this result. But the other, and we know that Geisenheim is something special because we looked at in France about 21 places and have this result over all the 24 places. Okay, you ask in between if you have questions. So when we look now at the field trials, we really found significant with only Geisenheim not, but in all the other places we found significant more plant growth promoting microorganisms when we sprayed biodynamic preparations. And the next is what I told is the time series. So the chief from the laboratory told us, okay, if you want to look, if you expect something like an inoculation with bacteria and fungi in the soil when you spray preparations, then you must look at the time series. And the single groups it's different and it's a lot of, how do you say, Schwankung. But so it's a lot up and down there. We checked this on 3 places in France and 1 place in Germany. But when we take the mean of all, we see a clear that it goes up to the 8 weeks, so 2 months, and then after 11 weeks or 15 weeks, it goes down again. And that's what we expect when we inoculate soil with bacteria and fungi. So this
You mean there's a lot of variability, yeah? A lot of variability in the samples, but when you take the average, then you see the direction. It goes up and down.
Yes, that's it. The variability. This was the word I searched. If you only look at the single groups, there you see a lot of variability, but in the mean you see the direction clear. Yes.
Yeah.
Okay. So when we look at the time series, it's coherent how it's reacting. And we see also how long it needs. And we know from soil structure that we have an effect also about 8 weeks after spraying, intense spraying, we have effects. And then, yes, you have a question?
Well, yes, you're presenting a lot of interesting things. So, questions, they're coming like fungi, Okay. What about the period? I mean, what season was that? And did you see any diversification with geography? I mean, the distribution, the climate zones, or things like that?
Well, the climate zones, we only checked it in France and Germany. Mm-hmm. So I can't say something about climate zones.
Latitude, more or less?
Latitude?
40, 60. I mean, I tried to figure out the rain values, the year rainfall.
The year rainfall is about 500 to 700 millimeter per year.
Mm-hmm.
And the preparations we spray mostly end of March and beginning of April. So there we start. And on some places, we sprayed here only once in France on 3 places. We sprayed only once on manure, and we have this effect here.
Thank you.
Okay. So the next question was, and this was a question from the man we asked to a microbiologist. So he was really a specialist in this part of science. And he said, okay, now if we have an inoculation, if we put bacteria and fungi, we have a lot in the preparations and we put it on soil and we get more in the soil, then it would be interesting to look at the groups who have a lot of stems, who have a lot of in the preparations, more than 1%, and we look if these stems are more in the soil. Do you understand? So, and we checked this. So what's a lot of in the bacteria, in the preparations, is there more than in the soil. And this is like this. So if we have from the bacteria and fungi, we have a lot in the preparations, we have more in the soil. And we check this also when we check this with the time series, we also see not only in the whole unit, but also in the groups who have a lot in the preparations, we have more in the time series till week 8, and then it goes down again. So with the results we have clearly that with biodynamic preparations, we have a sort of inoculation with plant growth promoting microorganisms in soil. We produce this in a fermentation in soil, and then we put it on the soil, and it helps the plants against, yes, against stress and all this. And
Yeah, so the question is about the plant, because did you check it while it was growing? What was growing there? Wine or other
a lot of
plants like
things.
wheat or what?
It
What?
was wheat, it was wine. So in France, it was on 13 places it was wine, and then it was grain, and then it was lot of fruits on soil and agriculture. So on different cultures, really different cultures all over. And what we know from
Did you check
what-
it? Did you check the plant themselves or not? You just checked the microbiome of the soil,
yes, we checked.
not the
we
plants
checked,
themselves.
yes, we checked the whole soil. So you call this so a mix from the whole soil.
Batch sampling.
Batch sampling. Yes, we did this. But I will show you what we want to do in future. So this is only the first step. So we will continue there. And we know what I told, that an inoculation is very difficult. And sometimes it works very, very good and sometimes not. And the same we know about the preparations. Mostly they work very good, but there are some places where we don't get effects. And this is similar, so we understand it better when we see the biodynamic preparations also as a plant growth promoting inoculation. And the plant biodynamic preparations, they have still a very low concentration. And it's the question, how can we understand this? That it works like this. And the first point is that the development is in soil. And we expect, therefore, a very good adaptation. Very much better than normally if people put plant growth promoting microorganisms on soil. And the plant, I told it also, the plant is increasing bacteria and fungi in their rhizosphere who are good for the plants. And they increase them very specialized. So only the one who are interesting for them, they increase them. And what I told before is bacteria and fungi are active about 1%. And by steering in warm water, we expect that we activate the bacteria and fungi. So, and we have a biofilm. Biofilm, I don't know how to say it in English. Do you understand it in English? It's not only in water, it's not dry, but it's in something, like the preparations, like colloidal, so who give them a safe place, bacteria and fungi. And they are more effective there. And with this, it's, for me, the first time that we have a model where we can explain the effect from the preparations. And one friend of mine, after the talk, he said, "Oh, the scientist in me, he is very happy, but the biodynamic in me, he think, oh, it's only an inoculation what we are doing with the preparations." And I think that is not true, because how should I tell? What we are looking with this way for looking for the microbiome, we are looking at the part, only one part, and we are looking only the part of the living. And we have very much, how could I explain? There are much more other levels combined with the preparations, and we look here only at one part, at the part from the living. And the question was how to continue, and there I must check short. I show you. You see the development in soil and a good adaptation. We now look, before we put the preparations in soil, we look at the preparations, we look at the soil, and after, when we put it out from the soil, we look at the preparations and at soil, and we look how this is changing. And from the plants, it was only also a question of, if we look at the plants, so we look at the bulk soil, the whole soil, and we look directly to the rhizosphere, very close to the plant. And we take also the roots, and we take also the leaves. So this is what we are continuing with our research. And with the bacteria, how we activate them, we are looking before we are stirring and afterwards. This is the program we want to do. Maybe we make a break here. I'm not finished, but maybe it's good first to have a talk about this. And
what you actually said now is that you expect more advantages from the preparations, not only the bacteria and the microbiome.
yes.
And in order to check these advantages, you check also the quantity of the roots, of the leaves, of the plant itself, etc., etc. Yes, if I understood it right.
Yes. We expect more effects, yes, and we expect that the effect by the roots is much stronger than in the whole soil. You know, when the plants and the roots, they make more, the bacteria and fungi who are good for their growth, then we expect that especially the plant growth promoting microorganisms get higher from the plants, and there we expect more effect in the close roots. Yes.
Jürgen, did you also check the secondary metabolites activity in the root or in the leaves of the plants? And did you see any effect in that direction?
No, we did not this, but we have results. I will show you later. We have results from the preparations on secondary ingredients, and we will have a look there.
Thank you.
Additionally, when we talk about when I landed in university, it was about this bacterium that helps the plant to get the nitrogen from the air.
Yes.
How is it called? It has a name. Anyway, this bacteria, they connected it to the nitrogen, that the plant can take more nitrogen from the air because it has the bacteria in the roots or near the roots, etc., like the ligands, like the
mycorrhiza.
This family of plants. איך אומרים? לירון.
Mycorrhiza.
קטניות.
Legume is the family, but it works with mycorrhiza.
Yeah, they have these, in the roots, they have these bulbs with the bacterium, and it helps them to get the nitrogen.
Yes.
But now in this research, you say it's not only nitrogen, it's the whole feeling of the plant, yes, in the land, in the soil, yeah?
Yes.
That's it. You got it. So it's a whole feeling. It's a whole. So the plant can produce auxin and cytokinin and gibberellin and all the other substances, but the bacteria help them to do this. So this helps them to grow better. Yes. And the plants interact with the bacteria and fungi, so they do it also in interaction. Like you know it from the nitrogen, they do it also with other substances who are helping the plant to grow. And in science, they start not to look only at the plant as the plant, but the plant and the microbiome, the bacteria and fungi together, they are like a bigger organism. They call it holobiont. So this is the bigger organism, the plant together with the microbiome, and they also do it for human. And so this is better to understand what is acting there. Yes.
But in humans, the holobiome is inside the stomach, and in the plant, it's outside.
Yes. It's like this. At human, it's also for parts outside, but mostly it's in this, not the stomach, but the gut. So the most is inside there, but it's also an interaction between the organism and bacteria and fungi. And this is very important. So there are a lot of diseases if this is not working good. So I can show you if you are interested in a figure. There is very, very much about this. Okay. How long do we have time? We have taught half an hour now? Still left?
Yes.
Okay. Mm-hmm. Tim, you had a question.
I always have one.
Yeah.
Well, I understood that the preparation, they've got the small ecology, huh? So there is a balance between the microorganism, the microcosmos. So one of the directives on the use of preparation is not to mix with other things of the same kind. Well, it sounds good in the first instance, but for instance, if someone is going to introduce EM, effective microorganism, or mycorrhiza, is it something which really is going to disturb the whole ecosystem that you're talking about?
I don't expect this. I would put my energy in biodynamic preparations because I know they are working, and I saw still now not good experiments about EM. And I had just on a conference, 2 weeks ago, a colleague who made a lot of work with also the, how do you say, it's regenerative Landwirtschaft, regenerative agriculture. And they also spray different things, and they tested it, and he was very frustrated because afterwards in a talk he said, oh, it would be better to put this time in biodynamic preparations than in the spraying. They had no effect, also not on the roots. So he was frustrated. But I can't say, sometimes maybe it works, and also with compost tea and things like this, there are different things you can use, and maybe something is also working. But still now, I know it from the biodynamic preparations, and I saw it again and again in the soil that the structure, if you have good preparations and you bring it out in a good way, then mostly you have it after 2 or 3 months, you have effect in the structure in the soil. And this is a way how the maçons in France, how they if someone was coming to them and said, oh, I want to change to biodynamic, they said, oh, wait, take a field and take one half with the preparations and the other half without the preparations. And when you see effects, then change your farm. And if you see don't effects, then let it go. And they changed, only everyone changed. So, this is quite interesting. But I can't say, I expect no disturb if you use something else, but maybe it's more interesting to put the energy in there than to spray 3 times or something like this. Okay.
Can I ask something?
Yes, of course.
In any of those treatments, did you check compost? The regular classic way of enriching soil and the root zone.
Mm-hmm. The compost preparations of compost.
He asked the normal compost.
Normal compost.
Yes.
We have several of them in Israel. We have only from wood chips. We have from cow manure and chicken manure, the classic ones, and we have sludge or biosolid compost.
We don't check the compost. I can show you later an experiment which is very known in Switzerland, where they check the compost and the compost together with the preparations. I can show you results there. This is really important. So
yes, this should be very interesting.
In praxis, in biodynamic praxis, we use both compost and preparations, and the experiments are only made focus on the preparations to look if the preparations have an effect. But in praxis, we need both the compost and the preparations, and we get better results then, of course. I show you the results later.
Yes, please.
Mm-hmm.
Thank you.
You're welcome. Okay, then we go further. You see it? Okay. Then we have, okay. Then we continue. Oh. Moment. Okay, now we look at the quality of the preparations. And these are results from France. And they are made from a working group in France, from the INRA. This is a well-known institution in France. And wait, there's going something miss. And I told you that the one, I checked it too quick because the time lag. I'm sorry. Okay, this looks better. Masson, he worked together with a group in France, scientist group, and he gave me these results. So I told you that, first we look. Here we have the microbiome, the networking from the microbiome, bacteria and fungus, how they work together in a natural system in the wood. And we see here the points are very close together. So this means that they are working very good together and very straight together. And if we go to prairie, to Weide, so if we go to agriculture, cereals, and to monoculture wine, when we go more and more in less natural situation, then we find that they are not so close connected with each other to work together, and we get small islands, and this makes the system less resilient. So in the natural system, in the wood, this is very resilient, and in the wine monoculture, this gets much more less resilient. And if we look now in conventional, so we have it more not so close together, and we have two islands. And we look at the organic, we see it closer together, and we look at the biodynamic, we see it very close together. And this is special samples we can see here, but we also can see it statistically. If you're interested, I can show you figures about this, but I don't want to show you too much figures to get you not overload with this.
Jürgen, are those populations or population distribution? What are those figures?
What was the question? Sorry.
Was it the population? Are you showing up the populations of the organisms in different agriculture methods? Or?
Yes, yes. This is the population of bacteria and fungi in different cultivation. One time is conventional, one is organic, and one is biodynamic. And in conventional, they are not working so close together. And in organic, they are working closer together. And in biodynamic, they are working very close together as we have it more in the natural system. You got it, or if you have questions? The networking is better and closer and stronger in the biodynamic preparation, with biodynamic preparations. And Vincent, he looked how does it go, how are the farmers, how made they their preparations. And 70% made the preparations not good at all. 50% made it good, and 30% made it very good. And if we look now, I must put it here. If we look now at the soils where the networking was bad and not good, what I showed you, then we had one part very good made preparation, but 3 parts very bad made preparations. And in the soils where you have a very good connection in between, a very good networking, you had 8 times more very good produced and bring out biodynamic preparations quality, and only a very small part, well, only one part bad made. You can see here's a correlation. The correlation is if you make the preparations very good, the chance that you get good effects in soil is much higher as if you made worse preparations, you bring them not good out and you have a bad quality. So it's important to have a good quality. And he has 7 points where he looked at the quality of the preparations from the farmers and how the preparations are made, if they are good humidity, if they are colloidal and things like this. He described it very good in a book. It's now in France and in German, and I expect it comes also in English. I show you the book later. Arbeitsbuch Biologisch-Dynamische Präparate. It's from Vincent Masson. But you also can see it on the website from them. And the next point is the storage, that you store it good. And the next point is the water, that it's slightly acid and not too much lime. And a good heating from the water, not higher than 35 degrees, but also heating the water and things like this. And the dynamization, how you steer the preparations. And afterwards, how you spray the preparations. And in the end, the timing. So it's necessary that you have a temperature about 11 degrees in the soil when the plants begin to develop in spring, if it's still warm in the end of the year. So he describes all this. And for me, it was important to show you that we get higher effects, okay, but maybe I continue. If we make it in a very good way, the preparations. Now the question was also from you, how is this with the secondary plant substances? And we have an experiment about pumpkins in Litauen. And there we have a fact about the soil enzymes, they get higher and the yield gets higher. But I wanted to go to the secondary substances with biodynamic preparations. Oh no, I have also, but this is maybe now too much. I go through. This is too much now. I want to go to this. You see, we have 3 cultivar from pumpkin, and we have 3 years and 4 replications. So we have a very good database. And we see when we have horn manure and horn silica together, the amount of secondary ingredients gets nearly to the double in 2 of 3 cultivar. And in the 3rd cultivar, we also get significant effects, but not so high. And what I told you in the beginning, so after spraying wine on the wine leaves, in the wine leaves, we see that the structure of secondary substances is changing after 6 hours. You see it on one place here, and you see it on another place here. And after 11 days, you just get more, also the same effect, and you see it also in the amount of secondary substances. So you see, if you have a plus, this is the different without against with preparations on 2 places, and you see the different secondary substances. You see this in the leaves, and you see the same in the berries. In the berries, much more. One, you have in the other direction only at one substance, but overall, you have much more secondary plant ingredients. Okay. And this was a question, what is changing if we not only change preparations, but the combination from preparation with compost? And this is what we see in experiment in Switzerland. And we see here the effect after 45 years. And the green line is the line biodynamic normal amounts of manure with preparations and compost. And blue is normal amount, the same amounts of Dünger, of manure. The same amount of manure, of nitrogen and things like this, substances, but in organic without compost and without preparations. And you see the line is all over the time higher in biodynamic against organic. And yellow, it's a DOK. Okay. And it's very new results about this. So it's maybe I try. No, I don't have it here. No, I don't have it. I thought maybe I have a picture about the literature of the. Okay, but it's from the DOK. It's new results about the time over 45 years. So here we see the effect, the combination from compost and preparations. And here we see also the effect from compost and preparations and the microbiomass against organic. Green is biodynamic, preparations with compost, and here organic. And then this is mineral. And red is a combination from manure and mineral fertilizer. And yes, it's too much. We look here in the structure. We see the structure in the soil is the best in biodynamic. Then there is organic. And then we see mineral and conventional. Okay. what do we see now? We see that we have climate change, we have the problems in nature and for plant growth. One part we can work and help and reduce a little bit about this is with the biodynamic preparations as an inoculation from plant growth promoting microorganisms in the soil. The second part is the biodynamic preparations, they make more secondary plant substances. And this is also helping against growing stress. This is the main, and much more. So this helps the plants to grow. And the third one is in the DOK trail in Switzerland, so the effect from biodynamic preparations together with compost, we see higher soil carbon, we see a better soil structure. And the CO2 equivalence I didn't show you. Did I show you? No, I didn't show you. Wait. Yeah, it was too quick. I show you. So the effect on CO2 equivalence in soil, we see carbon storage, biodynamic is the best, and then is coming organic. Methane, there is not a big difference. In nitrous oxide, it's in German, it's Lachgas, it's laughing gas. How do you say? It's really important for the climate. And there the effect in biodynamic is much better, though the bacteria and fungi in the soil are producing also this. This is much better, or this is better in biodynamic against organic. And if you look overall, the effect for climate is the best in biodynamic. And biodynamic is compost with preparations. And then there is organic. And then there are the conventional. Okay. This is what I wanted to show you.